So, what if I told you that the secret to understanding the food on your plate is actually hidden inside a high school biology exam? Today, we're not just going to study for a test. Oh no, we are going on a guided tour of nutrition, and we're using an actual exam paper as our map. Let's dive in. See, this isn't about just cramming facts. This exam is basically a masterclass in biology. It takes us on this incredible journey from a tiny plant cell all the way to our own digestive system. It's our blueprint for understanding how life itself is fueled. So where does our story begin? Well, it doesn't start on a dinner plate. It starts outside with these silent green engines that literally power our entire planet. Plants. We're going straight to the source. I want you to think about your last meal. An apple, a sandwich, whatever. Where did the energy in that food really come from? I mean, if you trace it all the way back, you'll find it all starts with one incredible, planet-wide chemical reaction. Photosynthesis. You know it, you love it. This is basically nature's most important recipe. On the left, you've got your ingredients, carbon dioxide and water. You add a little bit of sunlight, and then boom. On the right side, you get glucose, that's pure chemical energy, and as a little bonus, the oxygen we're all breathing right now. So yeah, you've got light, water, and CO2. But the real magic, the real MVP of this whole process is chlorophyll. And here's a super cool detail. Right at the heart of every single chlorophyll molecule is one atom of magnesium. Think about that. No magnesium, no chlorophyll. It's a tiny little mineral doing a planet-sized job. And all of this incredible chemistry is happening inside a leaf. But it's not just a leaf. It's more like a perfectly designed, solar-powered factory. It's an absolute marvel of biological engineering. Just look at this cross-section. That top layer, it's jam-packed with tiny solar panels, the chloroplasts, all angled perfectly to soak up the sun. And then the layer underneath is kind of like a ventilation system, full of these little air pockets that let CO2 in and oxygen out. It's just an incredibly efficient design. So you might be thinking, okay, if a plant has all the ingredients it needs, does that factory just run full throttle 24/7? Well, not quite. The whole process can actually be held back by something called a limiting factor. You can think of it like a bottleneck on a production line. This data, straight from the exam, tells a really clear story. Check out that top row. When there's low CO2, adding more light does absolutely nothing. The process is bottlenecked. But as soon as you pump in more CO2, the rate just jumps up, especially in high light. It's perfect proof that you need all the right ingredients in all the right amounts to really get things cooking. All right, so the plants have done the hard work. They've made the fuel. Now, how do we get that energy out? Well, our bodies have a specialized demolition crew just for that job. And that crew is made of enzymes. Now, this right here is the textbook perfect score definition. An enzyme is a protein that acts as a biological catalyst. In plain English, it just means they make chemical reactions happen millions of times faster than they normally would, and they don't even get used up in the process. They're like little biological superchargers. You've probably heard the classic lock and key analogy, right? The food molecule, the substrate, is the key, and the enzyme's active site is the lock. That key has a very specific shape that fits perfectly. And the key scientific term for that perfect fit, the one that'll get you the points on the exam, is complementary. Now, enzymes are amazing, but they're also, well, they're a little bit sensitive. They have a perfect temperature and a perfect pH where they work best. So what do you think happens when things get a little too extreme? If the conditions are wrong, say it's too hot or the pH is off, the enzyme denatures. The bonds that hold its shape together literally break. The active site changes shape, and suddenly the key doesn't fit the lock anymore. And here's a super crucial point. Enzymes are not alive, so they can't be killed. The word you're looking for is denatured. Speaking of creating the perfect conditions, we have to talk about a real unsung hero of digestion, bile. It does two critical jobs. First, it neutralizes that super strong acid coming from your stomach, which creates a safe, alkaline environment for the enzymes downstream. And second, it emulsifies fats, which is a fancy way of saying it breaks big blobs of fat into tiny little droplets. This gives the lipase enzymes way more surface area to attack. Okay, so we've covered a lot of the what of biology. Now let's shift gears and talk about the how. Like, how do we actually know all this stuff? We're going to use our exam guide to start thinking like a scientist and crack the code of how experiments work. So here's a classic scenario, straight from the exam paper, but it feels like a real-world problem. A company says they've got a new drug, a lipase inhibitor, that's supposed to stop you from digesting fat. They've run a tiny trial. Now our job is to put on our scientist hats and analyze their data. Okay, let's look at the data together. At first glance, the inhibitor seems to be working, right? The person who took it has lower blood lipids. But hang on a second. Look at that third person. They had no inhibitor, but their lipids are also low, and they had the same side effects. All of a sudden, the picture isn't so clear, is it? And this right here is the very heart of scientific thinking. You have to be able to argue both sides. The argument for the drug is that, yeah, it seems to lower fat absorption, but the argument against it is just overwhelming. I mean, the sample size is tiny, the data is all over the place. One person's result could be a total fluke. There is no way you can make a solid conclusion from this. No way. And that leads us to probably the most important question a scientist can ever ask. It's not, am I right? It's, how can I improve this? How can I be more certain about my results? So if we wanted to fix that flawed drug trial, what would we do? Well, you'd have to control for things like diet and exercise. You need a way bigger sample size to get reliable data, and you would absolutely have to be on the lookout for weird results, anomalies that could throw off your entire conclusion. Okay, let's pull all of these different threads together. We've gone from photosynthesis to digestion all the way to experimental design, all by using this one exam paper as our guide. So, what have we really learned? We've learned that these aren't just separate topics in a textbook. No, they're all chapters in one single incredible story. It's the story of how a ray of sunlight travels 93 million miles just to become the energy that is powering you right here, right now. Because ultimately, just knowing the facts is only half the battle. The real power comes from understanding how we know what we know, how we ask questions, test ideas, and challenge assumptions. That is the true heart of science. So the only question left is, what biological mystery are you going to unravel next? Thanks for joining this explainer.